to our first video looking at histology and or tissues. And so in lab, we've been spending quite a bit of time looking at some histology, talking about the different types of tissues that are there, and trying to make those connections of understanding uh, for how are our organs organized. We know that it takes two or more tissue types to work together to do a common function or purpose or goal in order to be considered an organ. But what does that really mean? So in this video, we're going to kind of review those epithelial tissues, the connective tissues, and we're going to spend a little bit of time diving into the muscular, at least introducing to you the muscle tissue and the nervous tissue, and then end the video with a little bit of wound repair. So if you get a cut, how do those tissues and cells kind of all work together to produce healing? So with that said, let's go ahead and let's jump into looking at this first video, which is going to be all about epithelial tissue. The second video will be about connective, and the third will be about everything else, kind of lumped into one. So with that said, we know that there are four general tissue types, all right? And those four general tissue types are listed there on the screen in front of you. And so we should be reminded that those four general tissue types are epithelial, connective, muscle, and nervous. And I cannot stress this enough. These four are the general tissue types. All right. General meaning less specific. All right. When we say epithelial, what do we mean? So when we say connective, what do we mean? What kind? So these are very general classifications or organizational terms that we have. So let's go ahead and let's spend some time looking at epithelial tissue. I'm actually going to be doing this one a little bit more differently than what you might be anticipating. So general characteristics of epithelial tissue. All right. First of all, epithelial tissue is avascular. Right. Now, by avascular, we mean that it lacks blood vessels. So there is no blood vessels that are supplying needed nutrients and oxygen, and there are no blood vessels that are removing metabolic waste, such as carbon dioxide, ammonia, other nitrates, from these tissues. Right? So that's number one that you need to really keep in mind when we talk about epithelial tissue, is that it lacks blood vessels. It is avascular. Now, this is where connective tissues will come into play. So when we get into the next video, you will notice that connective tissues are vascular. And in fact, epithelial tissue very often relies on the vasculature of connective tissue to go ahead and supply nutrients and oxygen and remove metabolic waste. Another key characteristic of, or general characteristic of epithelial tissue is that they are lining. Right? It lines or covers an area. Very often, I'll use the case of or I should say I'll use the example of what we call proximal convoluted tubules in the kidneys. All right, these tubules are circular. All right, so if you do a cross section, you can kind of look into them, and then surrounding them Surrounding this, you actually have these cuboidal cells that are lining the tube itself. And then, so in this case, these epithelial cells, these cuboidal cells, are lining the opening to this tubule that we see right here. And because we're looking at that hollowed space, this is what we define as being a lumen. All right. A lumen, I'll write it over here just in case you can't quite see that. A lumen is simply a cross-sectional view of a vessel or duct. Right. So in this case, if this was in the kidneys, you'd be looking at the proximal convoluted tubule. Right. A tubule is a vessel, right, or a duct. And so when we talk about a lumen, we'll see that very often. We've looked at that in the lab, and I've used that term. Right. Another characteristic of epithelial tissue is that they have apical and basal layers. You have an apical layer and you have a basal layer. All right. The apical layer is what is pointing or exposed to the outside, right, or to the lumen of a vessel. The basal layer is directly attached to the basement membrane. And so all epithelial tissue also contains a basement membrane. And so if we look at my example up here, all of these cells would actually be in touch or in contact with a basement membrane. And so this would be the basal surface. And then this surface over here that is opening towards the lumen would be the apical surface. That would be the apical surface. Another way of looking at this is, here's my basement membrane. Right, remember, another term for basement is basal. Right, same thing. Right. And then sitting on top of my basement membrane, I would have my cell or cells. Like such. Right? And so while this surface is the basement membrane, this edge that is now being exposed, that would be the apical surface, right, or the apical layer. Right, so these are just some of those general characteristics that we see for epithelial tissue. Now, what about identifying? What about the organization of epithelial tissue? Well, I'm going to go back to basically an overview of what we talked about during the lab setting and just remind you that we have these various categories of selection that you can pick from that define and describe the epithelial tissue. So we've got we have a general column that we can pick from. We have a specific column that we can select from. We have an arrangement column that we can select from, and we have trying to think of the term that I'm looking for. We have a characteristic column that we can choose from. And so when we're going through and trying to name our epithelial tissue, we're basically selecting one term from each of these columns. Now, under the general term, right, we only have one term, epithelial. Right? This is our general classification. Right? All of these tissues and all of these cells that we're going to be looking at all fall under the general category of epithelial. Then, underneath of the specific, all right, another way of looking at the specific is what is the cell type? Right? Specific tissue, or what is the cell type? Well, we have squamous, we have cuboidal, we have columnar, and we have transitional epithelium. Right? So this would be the second category, or the second column, for which you go ahead and you select from. And then we have the arrangement column. Right? We have the arrangement column. And so under the arrangement column, we have, for squamous, we can have simple, or we can have stratified. For cuboidal, we can have simple, or we can have stratified. For columnar, we can have simple, stratified, or this term, pseudo stratified. And then we have transitional, which is always stratified. I'm going to actually put that in gray because even though it is stratified, we never say stratified transitional. It's understood that transitional is stratified. Right? So it's an unspoken term that we have. And then under the characteristics, we can have, for squamous, right, for stratified, we can have keratinized, or we can have non-keratinized. And then under columnar, we can have ciliated. So the way that this little chart works basically is, let's say you've got yourself a squamous cell. Right? Well, if it's stratified, then we need to also select whether it's keratinized or non-keratinized. Non-keratinized are things like where you find stratified squamous lining the esophagus, or lining the uterus, or lining the vaginal cavity. Keratinized stratified squamous would be the skin. Right? And I think we've touched on that difference there a little bit, and we can touch on it a little bit more. But you basically pick from each column. So keratinized stratified squamous epithelium, right? Or simple cuboidal epithelium, or ciliated. Actually, it would be pseudostratified ciliated columnar epithelium. We kind of reverse that for some reason when we're dealing with columnar. And then transitional would just be transitional epithelium. So on the exam, if we're asking for the general tissue type, it would be epithelial. If we're asking for the specific tissue type, it would be stratified cuboidal epithelium. If we're asking for the cell type, it would be squamous, cuboidal, columnar, or transitional. All right. If we're asking for arrangement, it's either going to be simple, stratified, or pseudo stratified. And so this kind of gives you that again that visual representation about what it is that you are expected to be able to know and identify as far as from an organizational standpoint. Now, what follows here are some images just to kind of help point out what you are looking at. So again, on this slide, this here is actually the lung, which is the organ, right? But it's simple squamous epithelium because the squamous cells are arranged in a single row. Right? These cells are arranged in a single row. Now remember, sometimes they bunch up, especially in the lungs. So when the lung tissue is relaxed, it's like a hair scrunchie. They come together, and so it might look like they are stratified, but these are actually indeed all simple. As opposed to something like this, this would be non-keratinized stratified squamous epithelium. How do I know it's non-keratinized? Because these cells on the apical surface, this would be the apical surface, right, have nuclei. These are all living cells. Right? It's stratified because this whole area right in here, right, are multiple layers, one on top of the other, of squamous cells. And then if you look right down this area, right, you can actually start to make out the basal portion of the membrane or of the layer. Right? So this would be non-keratinized stratified squamous epithelium because, again, all of those cells are living. Keratinized is going to look very different. Right? So this is thick skin, this is thin skin. This is something that is in Lab 8 that we've taken a look at. Right? If you look at all of these cells here, this would be the stratum corneum. It's the outer layer of the epidermis, right? And all of these cells here are non-living. This whole layer right here is non-living, and it's non-living because it has been keratinized. Right? This is what gets sloughed off. This is what gets removed. Right? But if you look deeper down in this layer, because this is all stratified squamous. This is all stratified squamous, but if you look down here, these cells, if you were to zoom in, actually have nuclei. Right? So those cells are still alive, and it's as they get closer to the apical surface that these cells gain the keratin that ends up leading to their demise. Right? Same thing over here. If you look at this layer of tissue right here, right, that's dead cells. Right? It actually looks like it's starting to come off in sheets. Right? This is keratinized. This is thin skin. Right? So you're only looking at this area right here. Right? But down here, you can clearly see that you've got cell nuclei. This here would be the apical surface. This would be the basal surface down here. Right? So that would be keratinized stratified squamous. This is simple cuboidal. Right? Again, just looking at this area right in here, right, you see one layer of cuboidal cells. But if we're looking over here, and it might be a little hard to see, this is actually lung. But if you look in this area here, right, this structure that you're looking at here is a terminal bronchiole. So this is actually what's bringing air down in. So you've got simple squamous all around here, but this is a terminal bronchiole. And terminal bronchioles are known for having stratified cuboidal. And so that's what you're looking at right there, stratified cuboidal. This area here kind of looks like it might be stratified cuboidal as well. And there's a couple areas up here that are stratified cuboidal. So again, one layer on top of the other. Columnar. This here is within the intestines, this is simple. Again, all of the nuclei are aligned in one row, right? But you can see these columns of cells. You can see the membranes really well. This would be the apical surface here, and what you can see really well in this picture is the basement membrane. You don't always get to see that good of a look, but if you look right there, you can see that dark little line. That's the basal layer, right, or the basement membrane that's coming right along this way. Right? Same thing over here. This is the apical surface, right? But in here, this is pseudostratified. So all of these cells are coming in contact with the basement membrane, which is over here. But if you look, it looks like that there are two rows of cells, because there's one row of nuclei here and one row of nuclei there, when in fact what's happening is these cells are squeezing real tight next to one another, and so that's displacing the nuclei. So pseudostratified ciliated columnar appears to be multiple layers, but in fact it is not multiple layers. It is all one layer. And then you've got yourself your transitional epithelium, which again is right here. This is the apical surface. This would be the bladder. Right, you can again really see nicely that basal layer right there. And again, what makes this transitional is that when you go from point A to point B, the shape of those cells transition. Right? So that as that bladder fills up, the cells closer to the apical surface condense, they compress. But when the bladder is empty, the cells closest to the apical surface are more tear-shaped. Right? And then I give you a nice little cheat sheet here as far as how you can really try to differentiate some of the different characteristics and features of epithelial tissue. All right. So this is where we're going to stop here for this video. Kind of review it. You should do pretty well with this, I think. It's kind of a review of what we did in lab. And any questions, let me know, as always. And hey, I'll catch you on the flip side.